Good morning, good afternoon, and good evening, everyone, from wherever you're joining us. And welcome to the Scientist.com and Axlead Drug Discovery Partners Inc. webinar titled Enhancing Clinical Predictability Through Human Disease Relevant Preclinical Models in CNS and Muscle Drug Discovery. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host. Today, we're fortunate to be joined by Dr. Koji Murakami and Dr. Tomonori Kitaura, respectively Director of CNS and Research Scientist in the Translational Research Business Unit at Axlead Drug Discovery Partners, Inc., who will discuss practical approaches to improve translational decision-making by integrating disease-relevant models, functional assessment, and mechanistic insight across CNS and muscle disease research. But just before we get started, we have a few housekeeping notes to help you get the most out of the event. First, this webinar is being recorded and will be made available to watch on demand shortly after the live event. Next, if you take a look at the interactivity panel on the right-hand side of your screen, you should see 3 tabs corresponding to chat, Q&A, and handouts. If you happen to experience any technical issues, please first try refreshing your browser, as this tends to fix most problems. But if you're still having trouble, you can reach out to us using the private chat function, and we'll help you to troubleshoot. We'd also love to hear from you, so please send us any questions, thoughts, or comments using the Q&A tab. And finally, you can also explore a few relevant resources in the Handouts tab to learn more about some of the content presented today. And with that, I'm very pleased to hand things over to the first speaker, Dr. Murakami. Murakami-san, thanks so much for joining us today, and the floor is yours whenever you're ready.
Hello everyone, my name is Koji Murakami, and I'm the Director of CNS Pharmacology at Axlead. Thank you for joining us today. I'm pleased to share our work and perspective with you. First, I would like to briefly introduce Axlead. Axlead is a Japan-based integrated drug discovery solution provider founded as a spin-out from Takeda and Teijin Pharma. We support global clients through pharma-originated research platforms, extensive drug discovery data, and experienced scientists. Today, we have around 400 employees, including approximately 200 scientists with advanced degrees. We also have more than 1,000 legacy projects and over 1.2 million available compounds. Axlead provides end-to-end drug discovery solutions. Our capabilities include screening, chemistry, DMPK, safety assessment, pharmacology-omics, bioinformatics, pharmacology, and animal models. By integrating these capabilities and disease expertise, we support drug discovery across multiple therapeutic areas, including CNS. Now, I would like to move to the CNS part of today's webinar. First, I will discuss why clinical success remains challenging in CNS drug development. Then, I will introduce the 3 major translational challenges and the solution we have developed to address them. The main message today is simple. We need stronger translational evidence to help increase the probability of clinical success in CNS drug discovery. CNS development remains one of the most challenging areas in drug discovery. Public benchmark data show that, on average, the likelihood of approval from phase 1 to FDA approval is only 6.6% across neurology and psychiatry programs. A major bottleneck is a transition from phase 2 to phase 3, where the success rate is only about 27%. This highlights the need to stronger translational evidence before entering costly clinical studies. So, what's the major translational challenges? We focus on 3 gaps: the human disease relevance gap, the functional biomarker gaps, and the behavioral translation gap. These 3 gaps will be the focus of the next section. The first challenge is human disease relevance. conventional animal models don't always capture human-specific biology and pathology. The second challenge is functional biomarkers. Traditional biomarkers may not fully reflect brain circuit activity or local neurochemical changes. The third challenge is behavioral translation. Conventional behavioral tests often rely on limited endpoints and may miss clinically relevant changes. Our goal is not simply to generate more data, but to generate more translatable data. To address these challenges, Axlead has built an integrated CNS platform that combines human cell-based models, functional readouts using EEG and microdialysis, and AI-enabled behavioral phenotyping. By linking disease biology, brain function, and behavior, we aim to generate stronger translational evidence and help increase the probability of clinical success. The first solution is our human-relevant in vitro brain model platform. Brain organoids can reproduce cortical-like structures with human neurons and astrocytes. We also use mouse brain slices reconstituted with human microglia to study human glia neuron interactions. MEA recordings allow us to measure synchronized neuronal activity and evaluate neural function. We can also model disease-related pathology, including protein aggregation and ischemia-like neuronal injury. Together, these models and readouts enable both disease and functional analysis in a more human-relevant setting. Here are several examples of disease-relevant applications. Human brain organoids can be used to evaluate protein aggregation, including A beta and alpha synuclein. We also use human microglia reconstituted brain slice models to evaluate microglial phagocytosis and human glial neuron interactions. In addition, our neural injury model allows us to measure clinically relevant biomarkers such as neurofilament L. These models support both mechanistic studies and pharmacological evaluation. The second solution is functional brain readouts. EEG measures brain circuit activity, while microdialysis tracks local changes in compound exposure level, neural transmitters, and protein dynamics. Together, these provide complementary readouts that helps bridge preclinical findings to clinical studies. This slide shows representative EEG readouts. EEG is widely used to both clinical and preclinical CNS research. In epilepsy, abnormal or epileptiform EEG activity can be observed in both humans and rodent models. Spectral analysis can be used to quantify changes in EEG activity across different frequency bands. Parameters such as the signal amplitude and frequency band power can serve as translational biomarkers, helping to bridge preclinical pharmacology and clinical outcomes. Microdialysis is another important translational tool. It enables direct modeling of local molecular changes in the brain over time. These measurements can include neurotransmitter, soluble proteins, inflammatory mediators, and compound exposure in the brain. As a result, microdialysis provides valuable information for PK/PD assessment and biomarker evaluation. The third solution is AI-enabled behavioral phenotyping. Conventional behavioral assays often focus on a limited number of predefined endpoints. However, clinically relevant behavioral information may remain hidden. AI-based approaches allows us to capture subtle and multidimensional behavioral changes beyond traditional measurements. This slide illustrates the concept. A compound may appear effective in conventional preclinical tests, but fail to show efficacy in patients. One possible reason is that conventional endpoints may not capture clinically relevant behavioral features. Our approach uses AI to identify hidden behavioral parameters that may show a stronger relationship with clinical outcomes. Here is an example of this approach. We first use AI to capture subtle multidimensional behavioral changes in animals. We then analyze these features to identify parameters that may correlate with clinical outcomes. Our initial results suggest an emerging relationship between animal behavioral parameters and clinical effect in humans. Combining AI analysis with CNS pharmacology expertise is important for interpreting these findings. We also see opportunities to integrate behavioral data with other readouts such as EEG and biomarkers. Now I would like to summarize the key message. We discussed the low clinical success rate in CNS drug development and three major translational challenges. We then introduced our corresponding solutions. The key message is that CNS drug discovery requires evidence across multiple biological layers. We need to understand disease biology, brain functions, neurochemical biomarkers, and behavior. To address these different layers, we combine human iPSC-derived disease models, EEG-based functional biomarkers, in vivo microdialysis, and AI-driven behavioral phenotyping. Axelis integrated platform connects these layers to generate stronger translational evidence. By combining human-relevant models, functional biomarkers, and behavioral phenotyping, we aim to help increase the probability of clinical success in CNS drug discovery. Thank you very much for your attention. If you are interested in discussing how these platforms may support your CNS program, please feel free to contact us. We would be happy to discuss your project goals and the most appropriate study design. Thank you again, and I look forward to speaking with you. Thank you.
Fantastic. Well, thank you, Murakami-san, for an excellent presentation. We'll now address a few questions on the CNS side. So if the audience has any questions that you'd like to ask, please feel free to submit them in the Q&A tab on the right-hand side of your screen. All right, Murakami-san, so your first question today is, many companies are developing organoid models today, and so what would differentiate your approach?
Thank you for your question. That's a great question. Two key features different our approach. First, we can generate organoid with high reproducibility, which helps reduce the variability often seen in organoid studies. And second, we can establish disease-relevant protein aggregation models that mimic important pathological features of neurodegenerative diseases. Together with MEA-based functional readouts, this enables reliable evaluation of both disease mechanisms and drug effects.
Fantastic. Thanks very much. Okay, somebody else has asked, if a client wants to evaluate a compound using multiple platforms, what would be the typical timeline?
Yes, thank you. The timeline depends on the study design and project objectives. However, in a best case scenario, where the major studies are conducted in parallel, we may be able to provide initial results in approximately 4 months. This can include human cell-based models, EEG, microdialysis, and behavioral assessments.
Fantastic. And I think the last question, of course, AI is really big right now. So AI-based behavioral analysis is attracting a lot of attention. What do you think is the biggest challenge in this field?
Yes, thank you. That's an important point. I think the biggest challenge is understanding what AI detected behavioral changes actually mean biologically. So AI can identify the subtle and complex behavioral patterns, but linking those patterns to disease mechanism and drug effects remains challenging. That's why we believe it is important to combine AI-based analysis with CNS pharmacology expertise.
Fantastic. Well, Murakawa-san, thanks so much for sharing your insights today. We'll now shift gears to hear from Dr. Tomonori Kitaura to cover the neuromuscular disease section. Kitaura-san, please feel free to begin whenever you're ready.
Hello, everyone. Thank you very much for joining today's webinar. Today, I'd like to talk about one of the biggest challenges in muscle disease drug discovery, improving the clinical predictability of preclinical studies. Using Duchenne muscular dystrophy, or DMD, as an example, I'll show how clinically relevant animal models and objective functional assessment can help bridge the gap between preclinical research and clinical outcomes. Here, today's agenda. First, I'll introduce the common traditional challenges in muscle disease research. Then, I'll focus on two key issues. The first is the lack of clinically relevant animal models. The second is the lack of clinically relevant functional endpoints. Finally, I'll briefly introduce our A-Booster platform for accelerating drug candidate generation. Let's begin with the translational challenge. We see the same issue across many muscle diseases, including muscle dystrophy, motor neuron disease such as ALS, SMA, and muscle atrophy. Many compounds show promising efficacy in animal studies, but those results often fail to translate into meaningful clinical benefit. Uh, what, why does this happen? I think there are two main reasons. First, the animal model may not fully reflect the disease seen in patients. Second, the endpoint used in preclinical studies may not accurately predict clinical benefit. So our goal is to address both challenges by combining clinically relevant animal model with objective functional assessment. Let's start with the first challenge, the lack of clinically relevant animal models. I'll use DMD as an example to show why choosing the right model is critical for improving clinical translation. Duchenne muscular dystrophy is caused by mutations in the dystrophin gene, resulting in the loss of dystrophin protein. Patients develop progressive muscle weakness from early childhood. As the disease progresses, respiratory and cardiac function also decline, eventually leading to premature death. Ideally, a preclinical model should reproduce this disease course. However, the conventional B10-MDX mouse develops a much milder phenotype. For example, grip strength and rotor rod performance show little or no impairment. Cardiac dysfunction appears much later, and these mice have a nearly normal lifespan. Because of this, the MDX model may overestimate therapeutic efficacy. To overcome this limitation, we established the MDX52 utrophin double knockout mouse. Unlike the conventional MDX mouse, this model develops early muscle weakness, motor dysfunction, cardiac impairment, and premature death, proving the disease cause that more closely resemble patients with DMD. This makes it more clinically relevant model for evaluating potential therapeutics. One important reason for this difference is utrophin. Utrophin is closely related to dystrophin and partially compensates for its loss. In healthy muscle, utrophin is mainly found at the neuromuscular junction and myotendinous junction. In DMD, utrophin is upregulated as a natural compensatory response. However, this compensation is much stronger in mice than in humans. As a result, conventional MDX mice develop much milder symptoms than patients. By removing both dystrophin and utrophin, the MDX utrophin knockout mouse develops a much more severe phenotype that better reflects human DMD. Next, I'd like to show you the characteristics of this model. The MDX utrophin knockout mouse show progressive deteriorations across multiple functional domains. We observe declines in cardiac function, muscle strength, motor coordination, exercise capacity, and survivals. Unlike conventional MDX mice, these changes appear early and continue throughout life. Overall, this progression closely resembles the clinical course of DMD, making this model well suited for evaluating potential therapeutics. Here are some representative data from our laboratory. Even at an early age, MDX utrophin knockout mouse already show clear functional impairment. For example, we detect early diastolic dysfunction by echocardiography, reduced grip strength, and impairment motor coordination in rotor-rotor test. These findings demonstrate that both cardiac and skeletal muscle dysfunction develops much earlier than in conventional MDX mice. This early and severe phenotype allows us to evaluate therapeutic effects more efficiently and with greater clinical relevance. Now, let's move on to the second challenge. Even if we select the right animal model, another important question remains. Are we measuring the right endpoints? To address this, I'd like to introduce two translational functional assessments, TORC and CMAP. Traditionally, preclinical efficacy has been evaluated using structural and behavioral endpoints, such as muscle mass, histology, creatine kinase, grip strength, and treadmill performance. These endpoints are variable, but they don't always predict clinical benefit. A good example is myostatin inhibition in DMD. In animal studies, myostatin inhibitors increased muscle mass and even improved grip strength. However, these improvements did not translate into meaningful functional benefits in clinical trials. This tells us that larger muscle don't always mean better muscle function. That's why we include TORC, which directly measure muscle contractility, and CMAP, which evaluate neuromuscular function. These assessments provide objective functional evidence that complement conventional endpoints and strengthen confidence in clinical translation. First, let's look at TORC. TORC provides an objective and quantitative assessment of muscle contractile function. In this method, we electrically stimulate the muscle and measure the force it generates as joint torque. Because the contraction is electrically evoked, the measurement is much less affected by motivation or learning than behavioral tests. Another important advantage is its clinical relevance. On the right, you can see quantitative muscle testing, or QMT, which is widely used in patients with DMD to measure muscle strength over time. This means we are evaluating essentially the same physiological function in both animals and patients. In addition to maximal force, torque also measures fatigue resistance and recovery after fatigue, allowing us to assess both muscle strength and muscle quality. The second endpoint I'd like to introduce is CMAP, or compound muscle action potential. CMAP measures the electrical response of muscle following nerve stimulation, providing an objective assessment of neuromuscular function. On the right is example from one of our rodent studies. After sciatic nerve injury, CMAP decreased immediately and gradually recovers, allowing longitudinal evaluation in the same animal. On the left example from clinical studies. CMAP-derived measurements are widely used in patients with ALS and SMA to assess motor neuron integrity and monitor disease progression. Using the same physiological endpoint in both animals and patients strengthens the translational relevance. Together, TORC and CMAP provide objective functional evidence that complements conventional endpoints. So let me summarize. We believe that improving clinical predictability depends on two key elements. The first is selecting disease models that closely resemble human pathology. And second is evaluating drug effects using objective functional assessment across multiple biological levels. By combining clinically relevant models with multi-scale evaluation, we can better understand how a drug works and make more confident decisions before moving into clinical development. Thank you very much for your attention. If you're interested in applying these approaches to your own drug discovery program, we'd happy to discuss your project. Please feel free to contact us using the email address shown in here. And finally, before we finish today's webinar, I'd like to introduce another capability from Axlead, a booster. A booster is designed to accelerate drug candidate generation by starting from validated science rather than from blank slate. It leverages more than 1,000 pharma-originated legacy projects, together with pre-quantified compounds and data. This allows clients to start at the lead generation or even the lead optimization stage instead of starting from scratch. Combined with AI-powered DMTA cycle, a booster can deliver promising leads in about 6 months and support candidate nomination in around 1 year. Thank you very much for your attention. I'll be happy to answer any questions. Thank you.
Fantastic. Thanks so much, Kitaro-san, for a excellent presentation. All right, and with that, we'll get started with the Q&A session for this portion. And as a reminder to the audience, if you do have a question that you'd like to ask, you can submit it in the Q&A tab. All right, Kitaro-san, so your first question here is, does Axlead also provide histopathological evaluation?
Thank you for your question. Yes, in addition to functional assessment, we provide a wide range histopathological evaluation. We can score muscle fiber necrosis, regeneration, inflammation, and atrophy. And we also use Halo system for digital image analyzing to measure muscle fiber size and fibrosis. And for nerve tissues, we can analyze axon numbers and axon diameter. What is important to us is combining pathology with functional assessment such as torque, CMAP, and cardiac function. Looking at the changes together with functional data gives us a better understanding of how a drug works.
Fantastic. Thanks very much. All right. Next question here is, can Axlead design customized translational strategies tailored to a specific mechanism of action or therapeutic modality?
Yes, we can. At Axlead, we can design customized translational strategies for each therapeutic modality, including ASOs, and gene editing, and antibodies and small molecules. Depending on the modality and the drug mechanism of action, we recommend the most appropriate combination of in vitro assays, disease models, and functional endpoints, and pathological evaluations. These allows us to generate data that better support translation from preclinical studies to clinic.
All right, perfect. And I think we'll end with one last question here. Kitaro-san, could you just discuss what are the benefits of using this platform in drug discovery?
Yeah, thank you for your question. And the value of our platform is that it helps researchers make more confident decisions. We don't just look at whether treatment works or not. We also help researchers understand where the drug is acting and how those effects lead to improvement in overall motor function. So this allows researchers to evaluate efficacy data more accurately and provides stronger evidence to support go/no-go decision.
Fantastic. Well, thank you so much, Kitaro-san, for sharing your insights. It's really been a pleasure having you with us. A big thanks also to the audience for participating today. We now invite everybody to take a moment to share your feedback on the survey, which should pop up on your screen here. We do read all your comments and appreciate your input. Also, a quick reminder that a recording of today's webinar will be available soon, so keep your eye out for an email giving you access to that shortly. And if you still have a question, it's not too late. You can still submit it. There's a spot on the survey, and we will pass it along to the Axlead team following the event. So in closing, we hope you enjoyed this Scientist.com and Axlead Drug Discovery Partners Inc. webinar, and we'll see you again next time. Have a great day, everyone.